Mitosis is the process through which eukaryotic cells divide. So they divide to become two new cells which are identical copies or clones of the original cell. There's four main steps involved in the division. There's some other things which happen or occur in order to prepare cells, eukaryotic cells, for division, but these are the four steps of the actual division or the dividing of the cell itself. So the first stage is called prophase. In prophase, what we'll see is some centrioles start to move towards opposite poles of the cell. And what we'll see also is these little sort of microtubules, which we call spindle fibers, will start to form. Now, spindle fibers are little parts of the cytoskeleton, or similar to the microtubules which run throughout a cytoskeleton, and what they will do is they will help to split chromosomes up and spread sister chromatids across to opposite poles of the cell. Now, we will also see or have here the nucleus, but the nuclear membrane is starting to kind of dissolve and starting to disappear, at least for the time being. Inside of, I'll make these chromosomes a different colour. I'll make them a tiny bit thicker too. So inside of this nuclear envelope, which is starting to dissolve, we have some chromosomes in the form of sister chromatids. So they have divided. They haven't divided. They have replicated. So these chromosomes, which were linear, something like this, they're now looking a little bit more like this. The reason being is because they have copied their DNA. The DNA is replicated, so each chromosome now has two sets of that chromosome's DNA, because they're going to be split up apart across two new cells, and each of those new cells is going to need a full copy for itself. So what we have, and I've only drawn 4 in here because it will make life easier, but we have sister chromatid here. So this is just a chromosome that has had its DNA replicated, and these will start to kind of condense and coil, and we'll see them become a little bit more thick and a little bit more visible. So they'll be more visible under a microscope than usual during prophase. So remember also that there are not 4 sister chromatids. There would be 46 in the cell. I'll only draw 4 because it's a lot easier for me to only draw 4. But there would actually be 46 of these chromosomes, which have a double set of DNA. So when they eventually divide, each set will still have 46 linear chromosomes. Sorry, each cell will still have 46 linear chromosomes, giving it a full set, the full amount. Second stage is metaphase. So in this stage, what we've got is our centrioles and their spindle fibers. Our centrioles have gone to the opposite poles, and their spindle fibers will be stretching out across the cell. So our spindle fibers will be reaching towards kind of the middle or the equator of our cell. Now the reason is because in metaphase our nuclear envelope has completely dissolved, and what we end up with is we have now our sister chromatids align across the equator of our cell, what we can call the metaphase plate. So they align along the metaphase plate. Now spindle fibers, so these little microtubule-like structures, will spread out or reach out, and they will latch onto the centromere. So the centromere is a little central point which connects the two replicated copies of DNA which we have. So these spindle fibers will reach out, they will latch onto the centromere of the chromatids, the sister chromatids, which are lining up along the metaphase plate there. They'll latch onto the centromere. So in anaphase, which is our third stage of mitosis, in anaphase, what we'll eventually see is that these sister chromatids will start to pull. These spindle fibers will start to pull these sister chromatids apart, and will start to take a chromosome. Oops, I'll put the E on an anaphase there. They'll start to take a chromosome to an opposite pole. So what we'll see is this kind of thing occurring. So where they were latched onto sister chromatids here in metaphase, they've now started to pull each sister chromatid in half. So what we would have had originally was our sister chromatids like that. Now they've been pulled apart, and each is going to take a daughter chromosome. So we'd call one of these a daughter chromosome. Spindle fibers are going to pull to opposite poles, pull to opposite poles a daughter chromosome, so that each cell would eventually, in a human, have 46 individual chromosomes. I think that's one thing I didn't specify about this number here. Sorry, 46. This refers to human eukaryotic cells. So in other organisms, other eukaryotic organisms, this will be their method of division, cell division, but they may not necessarily have 46 chromosomes. They may have a different number. In humans, 46. So after anaphase, what's happening in anaphase is the sister chromatids have been divided, daughter chromosomes have been pulled apart by the spindle fibers to opposite poles of the cell, opposite sides of the cell. Now, our final stage of division is telophase or telophase. And what we'll see here is we can get an idea of the fact that we've got two cells starting to form. So a cell is actually being pinched. We have what is called a cleavage furrow. Start to form in a process which is called, I'll write it down the bottom here, cytokinesis. So this is the pinching of the cell in eukaryotic cells. In mitosis, cytokinesis is when the cell actually starts to be pinched into and divided into two separate cells. So what we'll see at this point is our centrioles will have done their job and kind of moved off to the side. We'll have a lot there in each of the new cells. But now, our nuclear envelope, our nuclear membrane in each of these cells is going to start to reform. And it's going to reform around these daughter chromosomes, which now are back to being linear form, and we would have 46 in this cell, and we would have 46 in this cell. And once this cleavage furrow has made its way completely through it and we're divided into two separate cells, we now have two identical daughter cells, two identical clones of the original dividing cell. So mitosis is constantly happening inside of our body to replenish or replace damaged or dead cells, and also to allow our bodies to be able to grow, create new cells so that our limbs can grow and our muscles can grow, for example. So those are the four key steps: prophase, metaphase, anaphase, and telophase, which make up mitotic division.